this pathogen doesn't belong to any of the Rebecca Lansfield group. So we talk about that. This is a so important human pathogen. But it's not a group A, group B, group C, group D. Why? Because there is no M protein. M protein, it is also an antigen. So this is the thing is important is we talk about so-called the antigen is a partial section, specific location of a bacterial cell. It is not the whole bacterial cell. And this is a good example which tells you a location of different antigen. So you have M antigen, Streptococcus pyogenes, that's why we can say group A strep. Plus membrane antigens, ribosome antigens, K antigens. So K antigen, lots of the time, we have E. coli, K88. That is from the K antigen. But more obvious, we talk all the time, is flagella. That's why we have H antigen and O antigen is LPS side chains. And that's something we want to talk about for the bacteria. It's very specific. Okay, so we talk about the antigen. Now we're going to talk about the antibody. So what is antibody? Well, antibody, obviously, it is a protein. So what it looks like. And you should be very familiar with the structure. The structure is right there. Go here, here, and go here, and here. It's a Y shape. So how are we going to talk about this? This long line, it's called H chain. How we say it's an H chain? Because the molecular weight is higher. It is 50,000 delta. Then you have this side one, we call it L chain. Why we say L chain? Why we say light chain? Because it's only 20 to 25,000 delta. Relatively small molecular weight. And they have to connect it. Like lots of the other proteins they connected. They need to connect it using S. So what are these? What is this called? What does S mean? Disulfur bonds. Okay, now we have an antibody. We need a place to recognize by the antigen. So where are they? It's usually on the top right here. For antigen to recognize and attach it because it's a variable zone. And then you have this long tail. So what's a long tail we talk about? This tail, we call it the FC portion. We call it a constant region. This is could be recognized by a macrophage. So this comes out with the next question. We talk about the antigen and antibody, but how they reacted with each other. It's a good picture for you. A very nice picture for you. There are 5 different scenarios will happen. The number one, which is right on the top, everybody knows, is precipitation. Antibody antigens reacted, they precipitate. Like the tubes, stay at the bottom. That's called precipitation. Okay, I need to talk this out. We want to talk about precipitation. So antibody antigen reaction, there are 5 different scenarios. The first one, precipitation. let's say they are, stay here. Instead of they just precipitated it, they could become this. We call it clumping. So what is that? Agglutination. So in the straight line across the picture, that's called agglutination. Because the antibody and the antigen have these characteristics for the reaction, we could developing an very simple immune kit to test the bacteria. Right here. That is called E. coli agglutination testing kit. You have a manufactured antibody, you have a control, then you have an antigen. The antigen is you test the bacteria. So you mix it. If you see something like that, we call it a clumping. Lots of dots, cloudy area, we call it a clumping. Why they can do it? Because it's caused by agglutination. Now what's the difference between agglutination and the precipitation? This is at the bottom. This is cross-linking. So that's very interesting. We talk about this. Okay. Other things. The third one, neutralization. Neutralization, basically, antitoxin. So we neutralize it. We mentioned already. Number 4, opsonization, right on the top. We talked already. Antibody and antigens, they reacted. They cross-linked. But the body doesn't think it's enough to kill them. So what we do? We bring white blood cell, engage it. We bring macrophage. Because when we bring the macrophage, it could be engulfed, already been combined antibody-antigen complex, and then they destroy it. Including all kinds of chemicals, lactoferrin, lysozyme, all kinds of things generate. That's called opsonization. So, opsonization, if you want to say very simple, is engulfed with macrophage. That is the key. Last one, complement fixing. So complement, you know that C1, C2, goes C28. That is a small immune-activated cell or the protein cell in the bloodstream system. They are just like a patrol. It's like a sheriff or the policeman just go in the body and to see, oh, where need help, I'm going to come in. When they see an antibody and antigen reacted, but they didn't end up with anything, they're going to come in to fixing the problem. That's called a complement fixing. So which means they're going to be enjoyed join the antibody-antigen reaction, and finally will cause the lysing of the bacterial cell. That's called a complement fixing. So this tells you the antibody-antigen reaction is not that simple. Lots of the time, its precipitation could happen. Agglutination will cause cross-linking, and then we can make a kit in the lab. And very ideally, we need a neutralization, especially for the virus. We have a picture there. But more often the time in the body, we activate the whole system. We bring the macrophage, we create opsonization scenario, and we also, lots of the time, need help from the complement. So we call it a complement fixing. So that's 5 different scenarios happens for the antibody protection of the host. So we come up with this topic is vaccination. We will be very briefly here. I don't want to talk about this one too much because in the United States, vaccination is kind of like a volunteer, is like not a requirement. So we want to give you some of the basic knowledge. So do you still remember what VACCI means? The VACCI means cow. So the vaccination, the first time coming from Edward Jenner in Great Britain. It's a cowpox, is that right? We mentioned about that. So that's vaccination where it comes from. It is a partial of the artificial activate immune system. Now, the key important thing here, what are the examples of the vaccine? We mentioned, listed 5 here, but there's more coming. First of all, is killed microbes. They could make polio and whooping cough. Bacteria has been killed. If it's a killed bacteria, that's not very effective. What this means, which means it's gonna be activate T cell lines, which means you need a booster. The most effective vaccine is attenuated. What means attenuated? It is a live bacterial cell. We do a little bit of treatment. Lots of the time is heat injured, which means they're still alive. The toxin is there. So what does the immune means? What does the vaccination means? We give you a low dose. We give you a low toxin in the body. Then to activate your immune system, tend to stimulate the host cell reacted with the pathogen and to create antibody. And hope it's going to be lost longer in your body. So attenuated is actually a treated live cell. That's why it is most effective. For example, saving polio vaccine and the very common one, MMR. What is that? Mumps, measles, and rubella. Mumps, measles, they should have been conquered for a long time, but they have recent news talk about the measles comes out outbreaks, but those are, we can say it's attenuated pathogen, which means it's like a new development. But traditionally we say mumps and measles should be gone, and the rubella should be gone too. This is required by everybody. The immigrants going to the United States, regardless how old are you, you need to take MMR. We just said attenuated vaccine is most effective because it's a live microbes. So usually this vaccine is one shots your lifetime. So MMR only one shot in your lifetime. There's no boosters. Purified antigens. What means purified antigens? Which means we isolated the partial section from a bacterial cell. It could be protein, could be nucleic acids, could be flagella, could be capsule. That's lots of the time we do that. That's where the whooping cough, meningitis, and we had streptococcal, pneumococcal. Those are coming from capsule. Toxoids, we mention all the time. DTAP, tetanus, diphtheria. What is toxoids? Toxic-like chemicals. Obviously, it is only activate T cell line. That's why you need a booster. Okay, with the technology developed, we have a genetically engineered vaccine. Let's say hepatitis. More recently, we have a COVID-19 vaccine coming from S protein. Is that right? That will be to inhibit the mRNA of the COVID-19 virus. So it's a brand new. It's not anything traditional we talk about here. So that's why it's not in here. But we just let you know there is a more developed biotechnology contribute for the new developments of the vaccines. So here is the vaccination, and we mention about this all the time, immunized hosts. So this is we talk about this in the exam too. During the infectious disease, this is a standard recommended child and adolescent immunization schedule in the United States, published in 2012, about a decade ago. So you can see all these listed, which is required to do. Lots of them are required. If you go to daycare or go to primary school, they need you to submit the kid's immune schedule. Okay, which one you did, which one you didn't do. You have to do their booster shots so you can enroll in the school. Immune response. This is something we want to say that you heard about antibody. We can treat the antibody. So for example, before the vaccine comes out, during the COVID-19 time, if you remember, how they do it? They get the blood cell from patients. And what they do? They centrifuge. Then once the blood cell centrifuge, they separate it into two sections. The bottom one is relatively red, because red blood cell on the top is light liquid. That is called serum. And the serum coming from COVID-19 patients, it is obviously is rich antibody from COVID-19, ideally. So that's why we call it anti-serum, which means it's rich certain antibody. And then we could do a serological testing to see how effective it is. Now how we did it. I want to tell you it's not easy to do. So just give me a very simple example to end up this section. So what happens is, in the old time, about 20 years ago, lots of people do this research. They need a cat, they need a rabbit. I'm not going to draw, so I'm going to draw like this. Yeah, like this. It's coming from a rabbit. Rabbit is an animal, experimental animals. So how do they do it? They go there to give a shot of Staphylococcus aureus. Now, of course, this shot of Staphylococcus aureus is heat injured. Otherwise, the rabbit is going to die. So heat injured. Want him to be survived. So what they do, they're going to feed up with carrots. All kinds of carrots have to be fed up. Let her to survive. And then what they do, sounds not very nice, they dig a hole in the ear. Then they get blood cell. When they get the blood cell, they do centrifuge, high speed. And what they get? Serum. And they hope this serum is rich anti-Staphylococcus aureus antibody. And once they get this serum, they do a serial dilution. They usually do a serial dilution, and you can see it. This is like a 20-fold serial dilution. Look at the titer. What is the titer? Reciprocal number of the dilution. And then they put into 96 well cell plate to react it with Staphylococcus aureus. Try to see is there any agglutination happens. And then they try to pick the first agglutination tube and find the titer number. And obviously the titer number has to be the smaller the better. We don't want it to be very high. Usually it needs to be at least 160 to 320, which means even you do a higher dilution, it still have a huge amount of the antibody. So that is a research people are doing all the time. However, it is difficult to do. What happened is they started to get a serum, but the titer is always very low because the amount of the antibodies is not enough. So they're going to be adding a booster there. Once they add the booster there, what they do? The rabbits cannot survive. It could be died, no matter what you are feeding the rabbits or any other things. So that is difficult to do. It took like 3 or 4 years to get the results. And that usually is a whole dissertation for a PhD student. And when you get the antibody, it is expensive. Usually 1 milliliter is $20,000. This is pretty much enough pay you 1 year tuition at WPU. That is why we do a lot of the work during the COVID-19 time. We collected a lot of the saline from the patients. But ideally, we never had public treatments to just give the patients shots of the antibody. The reason is it is difficult to do, and it took the time. Compared to you develop S protein, that is only a very short period of time. Remember, only like 3 months, 4 months we developed, and in secondly, in 2021, basically everybody can get the shots. Is that right there? But remember, the second shot is very severe side effects. You have a fever, you have some people have a vomiting. So this is the story about it. Okay? So that's we end up with this section. We have a last section on Thursday. We want to talk about the virus. And then we're going to do the review of the final exam. Then some of you want to do earlier, Friday afternoon, you can do the early exam. Okay?